I'm Daniel Levine, and this is the Bio Report.
Ovarian cancer remains one of the deadliest cancers affecting women, and it's expected to claim nearly 13,000 lives in the United States in 2025. Despite progress in survival rates, nearly 80% of patients are still diagnosed at advanced stages when the disease has already spread and it's difficult to treat. Imunon's experimental DNA-Mediated Immunotherapy is designed to deliver interleukin-12 directly into the tumor. A phase 2 study demonstrated that the experimental therapy, when combined with the standard of care, provided a 13-month survival benefit compared to women receiving only the standard of care. We spoke to Stacy Lindborg, CEO of Imunon, about the company's DNA-mediated immunotherapy, how it avoids the systemic toxicities that undermined earlier IL-12 approaches, and how it could change the treatment landscape for the disease. Stacy, thanks for joining us.
Great to be here.
We're going to talk about ovarian cancer, the poor outcomes and high recurrence rates, and Imunon's experimental DNA-mediated immunotherapy. Let's start with ovarian cancer, though, which can be challenging to catch because there are really no symptoms in its earliest stages. How significant a health problem does ovarian cancer represent?
Danny, it's a very significant health concern and really health issue. We know that 80% of patients are diagnosed in late stage, and a big part of that is due to the fact that patients present with nonspecific symptoms that are vague, such as bloating, pelvic and abdominal pain, perhaps they fill up when they're eating very quickly. Things that are frequently mistaken for benign conditions. And that added with the fact that there's no preventative screening tests that allow for early diagnosis really creates a health crisis for women when they're diagnosed. We know that late diagnosis leads to disease that's harder to treat. And many patients, as a result, can be somewhat debilitated by their disease at diagnosis.
How are people generally diagnosed?
So as we're talking about these nonspecific symptoms, it's often very challenging, but it can involve a combination of clinical evaluation, sometimes imaging. You can have vaginal ultrasounds or CT or MRIs. Sometimes it could be a lab test. CA-125 is a marker that's elevated in about 80% of epithelial ovarian cancer cases, although it's a lot more reliable in late stage diagnoses and less reliable in earlier stages. And then, of course, there's genetic testing that often is really critical, not only to the treatment, but also to diagnosis. So knowing if you have a BRCA mutation or are homologous recombinative deficiency.
I suspect one of the reasons that outcomes aren't better is because people are getting diagnosed late and being treated at later stages rather than earlier stages. But what existing therapies exist and how effective are they?
So the frontline standard of care is definitely effective. The challenge is that it really hasn't evolved in the 25 plus years since it became the standard of care. So I'll really be focusing my conversation today on the frontline treatment landscape. So these are women who have just been diagnosed, and it's the first treatment that they're getting for their cancer. And there's interesting context in this landscape. Number one, there's never been a trial that's had an overall survival improvement until our most recent OVATION 2 study. And number two, The standard of care, which is a platinum-based chemotherapy and surgery, is, while it's aggressively being pursued, we know that the majority of women will have relapses, and majority will also unfortunately succumb to the disease. So even as aggressive as these treatments are, and they do prolong life, the life is cut too short for many of these women.
What is the prognosis for someone diagnosed with the condition today?
The recent estimates, which have stayed pretty stable, is that a 5-year relative survival rate for a woman who's diagnosed with advanced stage ovarian cancer is currently less than 30%.
Imunon is developing an experimental therapy. This is IMNN-001. It's a DNA-mediated immunotherapy that encodes for the production of interleukin 12. How does it work, and how does it activate the immune system?
So, epithelial ovarian cancer is characterized by a prevalence of highly immunosuppressive tumor environment, and that's important to understand. And it's why we hope that Imunon can change this reality that the standard of care hasn't seen innovation for a long time. It makes immunotherapy a really attractive approach. So, Imunon 001, as you described, is an immunotherapy that's harnessing the power of each individual's immune system. And we know that this mechanism, the mechanism that we've chosen using interleukin 12, is very well established in the literature, dating back to really a key publication that was in 1989. Many attempts have been taken, around 40 clinical trials, to try to harness this very powerful anti-cancer fighting gene and cytokine. And so, why are we so hopeful? Well, because we've been able to dose at effective levels and really in a safe way, where others have run into toxicity challenges that have really terminated development of other products. And I'd say there are two really big factors that can cause why this would be effective. Um, and these are basically factors that would cause tumors to regress, and they are effector cells, which are T cells and NK cells. And the exciting nature of Imunon 001 is that we are harnessing the two major arms of the immune system, both innate immunity and acquired immunity. And we think that really is what is leading to the clinical data that we've seen that is gaining a lot of excitement.
Is this expected to be used as a monotherapy, or would you use it in combination with other medicines?
So, our product in our clinical trials and what we would expect, if approved, would be added to the standard of care. So, women receive cycles of chemotherapy, um, often before and after a surgery. So, women that receive what we would say is neoadjuvant chemotherapy, so prior to surgery, and we're seeing this actually increase. That physicians are seeing a benefit to shrinking the tumors before they go in surgically to remove them, and then you would follow with chemotherapy after surgery. Imunon is being added to that standard of care, and what happens is Imunon 001 is taken up, so it's administered through a catheter directly into the tumor microenvironment, and these nanoparticles are taken up by tumor and non-tumor cells alike, and it results in a local production, so safe and durable levels of IL-12, which we know is a powerful anti-cancer immune agent. And then we also see downstream cytokines like interferon gamma and other really important actors in the cancer fighting world that really result in a direct modification of the tumor microenvironment.
And is this expected to be a single dose administration, or would you need to do multiple dosing?
So in our phase 2 trial and what we're studying now in our phase 3 trial, in the context of a disease very difficult to treat, cancer that is almost always fatal, we're studying up to 17 weekly treatments of Imunon 001, or roughly half before surgery and half after surgery. And we're continuing with that same dosing protocol in our phase 3 trial.
And what's known about its durability? Is it something that would naturally be cleared from the body, or is this something you would have to somehow deactivate?
So the IL-12 expression, following, the administration into the intraperitoneal cavity really is expected based on our preclinical data and clinical trials to last for several days after the injection. And then you'll see levels that return near baseline level by about a week, which drove the dosing practice that we have and we've been exploring, which has been confirmed, with very clinically meaningful results. And it's our knowledge and belief based on our research that the TheraPlas components, the plasmid and the delivery polymer, are cleared passively through normal renal clearance because of their small size. So they're nanoparticle size. And so therefore there's no trigger needed for clearance or any other work that's required.
You briefly touched on safety before and some of the safety challenges past efforts to take this type of approach with the IL-12. But what do you know about the safety profile, and what has allowed you to keep this so localized?
Mhm. It's a great question and very important question for patients. So we have now, we're starting our fourth trial in the same patient population. So women who are recently diagnosed, and this is their first treatment that they're undergoing. And we see a highly favorable benefit risk profile in the clinic, these four clinical trials. And I think that really the route of administration and going into the peritoneal cavity is a central part of differentiating from our safety profile and those that have attempted to harness the power of IL-12 in the past, which were administered IV. So you had IL-12 being administered in the bloodstream, and you saw high levels because of with each of these different, the half-life and the dose that was required to make it therapeutic, resulting in immune-related adverse events, cytokine release syndrome, and even some deaths in clinical trials. What we observe and what we were extremely pleased to see in our very first trial in women that were newly diagnosed, we did a dose ranging study and we were assessing IL-12 levels and interferon gamma levels at different dose ranges. So starting very low, 36 milligrams per meter squared. And then we have now the fifth dose that we've studied, 100 milligrams per meter squared, that we're going to be taking into and are using in our phase 3 trial, and we believe would be the commercial dose if approved. And we actually measured a bunch of different translational biomarker data points in blood, and we also measured them in ascites, which is the fluid in the intraperitoneal cavity. And we were so excited and comforted to see, exactly as we expected, in the tumor microenvironment, we see a very strong dose response with IL-12 levels, interferon gamma, increasing multiple fold with increasing doses. So for interferon gamma, we're talking above 60-fold at the 100 milligram per meter squared dose. And with IL-12, we see above 25 fold increase. And what's really critical with that, if you look at those same women in their blood levels, we see incredibly low and almost no change across the dose levels. So really, we have a lot of confidence because of what we're seeing, not only in an efficacy perspective in what we can measure of the tumor microenvironment, but also the lack of changes happening in blood. And of course, it gives us also great confidence when we look at what we're observing in the tumor microenvironment, which is really taking what is known as a cold tumor environment because of the inflammatory nature of the disease and really making it a hot tumor environment. And from a design of the product, this is exactly what we expected to happen. And the downstream effects on the clinical side, we've shared very recently this year at ASCO and simultaneously published in the journal Gynecologic Oncology, is that this trial showed a 13-month advantage in overall survival in women who received Imunon plus the standard of care compared to those who were only receiving the standard of care. So we're talking about women living the median over a year longer than those who would have only been treated with the standard of care. And this is really a landmark outcome for this disease based on the fact that we haven't seen an overall survival benefit in any previous attempt.
What is the development path forward?
So we have a phase 3 trial that's ongoing, and I couldn't be more delighted at the momentum and the work of our team. You can imagine we have lots of metrics we're tracking just to see how we're doing, and I think this really speaks to the best and most exciting part of being in a biotech company. One metric I could give you is from the time that we got FDA approval to proceed with the trial and IRB approval to the first site activation, we actually accomplished this in half the time compared to the benchmarks of big pharma and really phase 3 trials that are reported on by global CROs that really conduct hundreds of these trials. And so we're moving quickly. We have multiple sites activated. We're steadily increasing that. And we're implementing an innovative design that's allowing us to focus on one aspect of our phase 2 data that I didn't actually touch on earlier, which is the high, we had a really large response in a subgroup of women that have HRD positive verification and diagnosis from biomarker lab test. So these are women with BRCA mutation and other mutations that would result in an HRD positive diagnosis. We saw an extraordinary outcome. So in the overall trial, we saw a hazard ratio of 0.7. And in this subgroup of women who were HRD positive, we actually saw the hazard ratio drop to 0.42. So we're harnessing that in phase 3. It's obviously very exciting to see an all-comers response like I described, but even more beneficial that we can think about how to really study this product and identify the most expeditious path to advance Imunon-01 towards potential commercialization.
And for a lay audience, what is the significance of the hazard ratio?
Yeah. So, again, going back, and a lot of the points that have really become very important to me come from my conversations with our PIs. These are physicians, men and women who have been treating patients with ovarian cancer and have dedicated their careers to this. They have continued to kind of point that while we've had a lot of attempts at the frontline space, of course, where we've seen most of the innovation for patients have come in, refractory second line, platinum sensitive, platinum refractory, the maintenance period. We've seen a lot of advances. But what we are, of course, really seeking is that first treatment to help put patients on a different course. And so that first hazard ratio of 0.7, again, when you look at that, you look to see, is the data mature? Have you been able to observe the median time, right? So if the patients that are in the trial, time to when you've observed half of the patients reaching an endpoint, and that's associated with the 13 months that we were talking about before. So if hazard ratios don't speak to you, I think often the time that's really given back to patients is a very easy to interpret metric. And what's interesting is with this subgroup that had this even really outstanding, even more outstanding response, I can't give you that metric because in the experimental arm, we still have half of the women in that group that are still alive and living well. So the standard of care, the median was observed at 37 months, but as of now, it's non-determinable what the median will be for this group, and that's great news for women living with this disease.
I want to also ask you about your platform technology for developing vaccines.
Mhm.
You develop nucleic acid vaccines for infectious diseases and cancer. The lead candidate here is a seasonal vaccine for COVID, but what's the threat for emergent viruses where vaccines don't exist today, and what's the potential to apply this type of technology to addressing that need?
Yeah, it's a great question. And we know that vaccines have been a cornerstone in combating a wide range of infectious diseases. And yet there are something on the order of, I think, 80 pathologic viruses since the 1980s, with fewer than 4% having commercially available prophylactic vaccines. So we know there's potential and there's need. Um, Placine is the second technology platform that we have, and it's actually a derivative of our TheraPlas technology platform. So we saw an opportunity to bring an improvement over traditional, which includes, also the more recent mRNA vaccines through exploring our ability to produce a DNA-based technology. And our business strategy seeks to capitalize on our competencies and our technology platforms. And I was actually on the board when we decided, with the board of Imunon, when we decided to undertake as a proof of concept study this COVID-19 trial. And we really thought it would be an efficient opportunity to do a real world trial against approved vaccines as a proof of concept. But it wasn't intended to introduce a departure for us from our focus, which is a cancer-based immunotherapy company. So, which is why following the completion of the proof of concept trial, we're actively seeking a partner to advance the development.
Well, how might the approach compare to more traditional vaccines or mRNA vaccines?
So why were we excited in it about it? Yeah. So what we understood in our preclinical data really kind of gave us this motivation. We fully expected that we'd see better durability of protection. We expected exceptional product stability and that there could be advantages in terms of speed to market because this proprietary technology has a flexible vector design that allows an expression vector to either be mono or multi-cistronic, meaning we could use a single or multiple antigens, and that would offer manufacturing flexibility and really speed if you had a need to develop a vaccine quickly. And in fact, what we've observed is just that. So on the stability side, there would be a clear commercial advantage to Placine. So we have stability up to at least a year in a 4 degrees Celsius setting, a month at room temperature, and at least 2 weeks in a 37 degrees Celsius temperature sphere. So it would be a simplified and really quite efficient supply chain as it would be required. And then we've also seen both in a host of preclinical experiments. We did human primate, rabbit, mouse, and then, of course, we've completed this phase 1 trial. And what we've observed is, in fact, we do see durability at up to 6 months, which is pronounced over what we've observed in the real world. And we see that that in fact, actually, at the 6-month timeframe, up to a threefold increase in the medium serum neutralizing antibody titers. And of course, what's really critical is that that's able to be done in a safe environment. That was the primary objective, the primary endpoint of this phase 1 trial was that to be able to demonstrate confidence that it's safe for human use, and in fact, we accomplished that. In addition to the ability to show that the vaccine is immunogenic across evolving pathogens.
One of the challenges in the past with DNA plasmid vaccines has been delivery. How much of a challenge is that for your vaccine?
Yeah, it really hasn't presented. I don't know if you mean supply chain. I've spoken to that
No, I mean
side.
delivery.
Oh, yeah.
Sorry. Delivery
Yeah.
to the cells.
Oh, okay. Yeah, it's not it has not been a challenge, at all. We didn't see any side effects, did not expect them, and we don't require any kind of special device. We're using a technology platform that, again, is evolved from TheraPlas and that we have a lot of experience with, and, it has not presented a complication at all.
Imunon is a public company. It's got a market cap of about $15 million. It had just under $5 million at the end of the second quarter. How far will existing cash take you, and what's the plan for raising additional capital?
So Imunon is very focused on bringing fundamental healthcare investors to partner with us that are going to be incentivized by the long-term success. We really believe that we have an unbelievable opportunity and we can transform the standard of care and bring true innovation to this frontline treatment landscape that's desperately needed. And um, if this process is done well, it takes some time to just like any relationship, but we've been actively and very aggressively working on taking the steps that are needed towards funding our trial with these like-minded investors. Um, you've seen, just like the rest of us, that it has been a very tough market. So there are definitely things in our control and things out of our control. But we're very, very pleased to see the market beginning to open up and to see the kind of deals that we're seeking to close that are actually being done. So, to ensure that we have the time it may require to really land one of these these substantial funds that would fund the trial, and with the people that we want to partner with, we've taken steps to conserve cash and align our needs with the critical needs with the available capital in hand. So we remain committed to funding this strategically, and we believe that we will, really be able to continue to keep the pace that we have set for this trial and fund it accordingly.
And do you think it's easier to raise capital today, given your size as a public company, or would it be easier to get a better valuation privately?
We've been able to continue to close bridge financing. So we've reduced risk for the company. We've chosen, again, we have a long-term goal of really choosing investors that are going to have the right incentives. And, there are other companies that have not been successful raising any kind of capital through any route. So I really attribute that to the strength of our data. I think our strategy of iterative financing is not that unusual, and again, we're really keeping our mind on the ultimate funding strategy, but we'll do what we need to, including bridge financings that, if required, to till the point where we can get there.
Stacy Lindborg, CEO of Imunon. Stacy, thanks so much for your time today.
Great to be with you. Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.